Well, good evening, and welcome to BIO212 Lab COVID-19 version. Dun, 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 blah. And we are here because you're missing lab on Friday morning, and I would like to introduce the material you were supposed to cover. And in doing that, I want to make sure that somehow we tie in the sort of current events, most of which you're now capable of understanding. Because of just some of the work we've covered inside this class already. So, sorry, I've got a cat running around on top of me here, and hopefully she won't disturb too many things while we're trying to get some stuff done. Okay, get up. Why'd you put you down there? Okay, so we're going to be looking at the lymphatic and immune surveillance system today. And so really what I want you to be thinking about is how and why do we actually even have this system? And you already know the answer to that question. Not that it really is a question. It's because, yes, you're a warm bag of salt water with other goodies that other organisms can utilize. And, of course, that makes you a meal. And a meal means many different things. But what you care about is the lymphatic system is a system that will enable two specific arms of surveillance inside of your body. There's a specific, meaning cell-mediated portion to it. And then there's, and by cell-mediated, I mean there are unique types of lymphocytes that are going to try to find what is specific about what is invading you, versus the non-specific cellular and non-cellular mechanisms that can try and protect you. And I'm just going to take this as an opportunity to sort of remind you of some of the things you already understand. Last week in class, I pretty much told you that after looking at the public comorbidity and co-mortality reports that are available through the CDC, I gave you a synopsis of the largest number of autopsies in place now. And we know that the virus tends to be targeting a specific population, doesn't mean anything right now, except that what I mean by that is there's going to be a lot more information being collected, and as the virus itself passes between host to host to host, it's going to pick up different characteristics. So even though we have an idea of how it's behaving now, it doesn't mean that's how it's always going to behave. That's the thing I really want to make sure you understand. Number one. Number two is crystal structure paper came out over the weekend, and I want you guys to wrap your brain around this idea. Let me get this here. It turns out it targets somebody we know. And by, I don't mean person we know. Who remembers what ACE is? Okay? So it turns out COVID, as it's co-opting what's going on inside of the cell, is actually, remember, we have A1 over here. A1 travels to where again? Do you remember? Where ACE is now going to convert it into A2? Well, it turns out it inhibits this pathway. So COVID-19 inhibits our RAA pathway, which makes perfect sense when you look at the actual mortality, or I should say mechanism of mortality. And even though it's exciting for me to say, look, you have an understanding now of how this works because of some of the material we've covered inside this class, it's probably more important for me to make sure that you understand that as you're going along, it means now that Mother Nature inside of you is going to have to figure out a way to see what's going on inside of those cells. Okay? And that's really what I want you to be thinking about when we're putting this lab together. Okay. Cat went away on her own. Woohoo! So how does it work? So this is you. It could be any one of you inside the classroom. And what I want you to see here inside green is this is your lymphatic system. And over on this side of our cartoon, this is to let you know that roughly all of the fluids in this part of the body are draining in on the right side of the venous blood return. And everything else in your body is being collected through a series of open circulatory structures that are feeding into this area of lowest pressure. And you'll see what that's going to be called in a few minutes. But I want you to take a look at the general idea of what happens. If you can imagine every single piece of tissue inside of your body where there is a blood vessel, and remember blood vessels look kind of like this, right? Let's go like this. Pick red. Right, red moves into a space and material is going to diffuse out of the capillary beds and they're going to be picked up inside of cells, and then whatever waste materials are there, right, they're going to be brought back in, and that's going to then return to your venous blood return. And remember, that's going to your right atria eventually, right? Okay. The piece that's new for our lecture right now is that you also now have these vessels, and I'm going to draw it traveling sort of perpendicular to this so it'll make a little bit more sense, where the interstitial fluid pressure inside this space will literally take the fluid in between the cells here and transport it into the lymphatic system. And this is what we mean by it being an open circulatory system. If these are all continuous vessels, the pressure pushes the space in between the cells open, and you'll see what I mean in a second. Now, before I move on, I want you to also see that the major structures of the lymphatic system aren't just these sort of conduit systems, but you can see it's the bone marrow. You can see its Peyer's patches are going to be found inside the digestive tract. You're going to see it's the spleen and the thymus. And today inside lab, you're going to be focusing on the structures and their characteristics as they function. And you're going to see that they have different levels of complexity. And I'm kind of hoping I'll make that clear in a few minutes. Okay. So the devil's in the pressure again. So here's that capillary bed, more nicely drawn for you here, where you can see blood's traveling into a space, and you have deoxygenated blood returning back to the right atria. And inside green here, you have more blown up for you here of the lymphatic capillary structure. You have these anchoring filaments that will, with pressure inside the space, push the endothelial cells open, and then that endothelium will now allow for the lymph, which is now present inside of that vessel. And like I said, I told you we would actually take a look at where they're draining, and here's the structure. If all of the fluid is traveling into the lowest pressure area, it's because it's traveling to the structure that we call the cisterna chyli. Okay, so all fluid, at least from the lower part of the body, okay, and the left arm, is going to travel here, and it will ascend through the thoracic lymphatic trunk, and will then deposit into here. The left lymphatic trunk here is going to travel into the left subclavian vein. Okay. Now, if we're descending from the head and the right arm, okay, you're going to travel in, and you're now going to enter in through the right subclavian trunk and then into the right subclavian vein here. So the purpose of me slowing down and pointing this out to you, and you can see down here they have the same sort of level of detail, is you can see that Mother Nature is figuring out a way to take fluids from the lymphatic system and then return them directly into the circulatory system. But you're like, well, what'd she do that for? You know, what is so significant of collecting all of that fluid? And you'll see why now. Okay, so before we actually get to the mechanisms, I want to introduce to you the first, probably, although very important, very simplistic structure that we call the thymus. And the first thing I'm going to teach you about the thymus is, look, it's deep to your sternum. It's superior and anterior to the heart. So if you're pointing to your thyroid gland now, you know not to make that mistake. It is a follicular-like structure where the maturation of T cells occurs inside the body. So materials move from the bone marrow, meaning the lymphoid T cells, which you're going to learn there will be multiple types. We'll have to figure out inside the lecture portion. But they come here to mature. They've been allowed to leave the bone marrow. They're now traveling to the thymus to mature before they will leave the thymus and populate peripheral tissues inside of the body. You know you're looking at a thymus if we're looking at histology here, and obviously we won't be doing a lot of histology while we're out of lab, but you can go to Google and look up thymus slides, and you're going to see you actually have a capsule standing on the outside. That's this piece here. And you can see you have this cortex surrounding a Hassel's corpuscle, which is inside the medulla. Okay, so the Hassel's corpuscle is inside the medulla, and you have this repeating follicular theme throughout biology inside AMP2. So if that's the case, let's get rid of that. Next slide. The spleen actually has multiple functions, and you remember the spleen is in the upper left region of the quadrants inside of the body. It's sort of almost retroperitoneal. It sort of sits in, pushed up by the stomach, and you can see that because you have a renal impression here, and you actually have a gastric impression here. So this is the renal part. It's pushing into the kidney. This is what's pushing into the stomach, and you can see it's richly oxygenated by that splenic artery and splenic vein, okay, removing materials from it. And you might recall from chapter 19, red blood cells come here to be reabsorbed or broken down. But what you don't know is that cells from the bone marrow, namely B cells, are going to travel here, okay, and they're going to take residency. Now, the interesting thing is, of course, by storing red blood cells here, your body keeps a reservoir of them should they need them for rapid utility. The purpose inside the lymphatic system, though, is for you to see, number one, here's a capsule. This is a sort of outer purple piece here. You have the red pulp, which is surrounding it, and you have this white pulp. Even though it looks purple, it's the white pulp, and has a central artery inside of it. And even though you can't make out the individual cells, Mother Nature is taking connective tissue, remember reticular connective tissue, and separating each of these regions by these structures that are called trabeculae. So what I'm doing is I'm going down the structures you're supposed to see in the slides or in the models, so that when I ask you to draw these or identify them on a quiz or something like that, you'll be able to do that. Now, you'll notice I'm not telling you really what the function of these things is. That's more for the lecture. But you should see that Mother Nature has now something with multiple functions inside of the lymphatic system. And what is the system that helps you understand what's going on here? Or what is the component to the system that helps you understand? What I'm looking at here, what you're looking at here in green, is this thing that's called a lymph node. Okay? And your body is collecting that lymphatic fluid, and it's passing through every single lymph node from whatever capillary bed it originated to that cisterna chyli. Okay, if it's going to the cisterna chyli, if it's draining down from the face, the head, the neck, it'll pass directly into the right subclavian vein, so it'll bypass the cisterna chyli. But in this case, okay, you're going to see that you have this capsule with trabecular-like structures. Should start sounding familiar, where you have a cortex that's surrounding a medulla. Okay, so you have an outer cortical portion here, but you have this sort of follicular-like structure, and you have the medulla inside here. And what I want to make sure you see is that whatever those fluids were, they are bathing over populations of cells. And in some instances, going from the outer cortex to the inner cortex to the cells of the medulla, till we finally have this area of lower pressure inside the hilum, and that's it's drawing materials through the lymph node. Okay, these cells are being bathed in that lymphatic fluid, so the lymphatic fluid can find whatever's inside of your body that doesn't belong there. Now, the follicular dendritic cells, the macrophages, over here you can see the macrophages and or dendritic cells. These are running around and presenting material. I'll explain that later inside class or lecture online, as the case may be. Okay? But the idea is this. By decorating the body with hundreds of millions of these, Mother Nature can take low pressure fluids, cascade it over all of the cells, and then move on to the next lymph node until we finally return all of that fluid back into your circulatory system. And from wherever it began to wherever it finally ends, every single B cell and or T cell that's occupying these spaces, along with the macrophages and the dendritic cells, they'll be able to identify that material that's trying to cause you harm. Okay? So a couple more slides here. Let's work our way through this. Let's get rid of that ink. Put that in there. Let's go down here. So what is it looking for? Well, every single one of those cells, now it's time to use your imagination. And I notice it's not spelt right. That's okay. Okay? Imagination. I'm sure that's going to help it. Okay. So what it's doing, of course, is whatever that particle is, and whatever the antigen is, and the antigen is just a fancy word to say that this is the thing that those T cells and B cells are looking for. Okay? And they're going to find specific epitopes that are unique to whatever those materials are. So how's it doing this? Well, remember a moment ago I said all those capillary beds are going to collect whatever those materials are inside of the fluid portion. Okay? And they're going to bathe through all of those lymph nodes until they finally get here. So by the time we actually return the plasma, okay, it's now inside your right atria, it's been mixed, but before that, it's been thoroughly screened by those T cells and B cells that are there. Okay? So if there's anything that could be found by those T cells or B cells, it will be found. Now, there's a host of other mechanisms, but we can't see them inside lab. What I want you to make sure you understand is that that lymph node is where all of the surveillance, the real sort of nitty-gritty levels of work are being done. Okay? And that's really important to wrap your brain around. So how are we then going to clean all of this out? Well, it turns out that not only do you have cells that will be able to identify it, if, say, a B cell is activated, let me put a pen on here. If a B cell becomes activated, it becomes a plasma cell, and that plasma cell will then release proteins into this liquid as well. And those proteins are what we're going to call antibodies. So let's get rid of that, and let's move to here. And I want to introduce to you what an antibody is. Now, there are 5 different classes of antibodies, but the one we teach you is IgG, because that's the one, if you have a flu right now, or you ate some bad food, and you rubbed your eyes, or you picked up something that's trying to kill you, Mother Nature's making a series of these proteins. And they're made up of 2 light chains. You can see light chain L and light chain right here. And you can see there's a variable region and a constant region. And then you have, because you have these 2 variable regions, you have these 2 heavy chains here. They're held together by these disulfide bridges. These identify other. So how's the killing done? Well, your immune system literally identifies the thing that's trying to harm you by randomly, and I do mean randomly, creating antigen binding sites that will identify that structure that's trying to harm you. Okay, it will bind to it, and this region down here is the stem region, okay, is then going to draw non-specific killers to actually remove whatever that pathogen is. So to finish off this sort of like get you to think about how to start working through these labs now that we're doing everything online till at least April 5th. I want you to go through the list of structures inside your lab packet. And remember, you still have to complete that lab packet for this next lab practical. I'm not quite sure what the lab practical is going to look like yet, but I'm working on it. And I'm going to try and find models online that look like materials we've used in the past, and I'll help you identify where to put the structures on that. I would ask that you try and get Kahoot on your cell phones, so if I put something together and I know it's going to be on there online for a period of time, maybe we can do something like that. But right now I'm going to work to create a path of progress to definitively cover the materials you're required to learn. So this is 19 minutes. This is our first attempt to do something new here. I personally don't believe in labs online, but if this is the best we can do at this point in time, we're going to make it work. Okay? So thank you for your attention, and I'll hopefully hear from you guys soon. I know somebody else has already reached out to me already to figure out how we're going to make this work. Okay? Bye. Oh yeah, wash your hands, stay away from your elderly relatives, and, just practice being smart.